for introducing me and for inviting me to give a talk in Cambridge. I'm really glad to be here, even though it's online. And please, I don't see any faces except for mine, which is even Paula has gone now. So please interrupt me and ask questions if anything is unclear. Otherwise, the talk will be more lively if you ask questions. And I don't know. Oh, there are more faces. That's great.
Also, if you want to put on the chat, I can also interfere and say, oh, there's some questions in the chat.
Yeah. Okay, that's fantastic. Yeah, so I always welcome interruptions. Otherwise, it's going to be perhaps a bit boring. So I mean, this is the theory of living matter group. And so I thought I put here on the title page also an essay we wrote about proliferating active matter, because in a way, one thing we would like to stress in this essay is that proliferation is a very special form of activity that leads to new phenomena that I think are worth exploring by the active matter community, essentially. And what I'll present today is an example of that, and gets into experiments that involve microbes and on the microfluidic scale. But in a way, the intellectual background comes from ecology and macroscopic ecology. The field is called island biogeography. So I'll introduce that, and it allowed me also to put this beautiful title page here of these islands, which is not exactly a true picture. It's partially fake, because 4 of those islands are real only, these 4 from the Maldives. And the other ones I just generated by copy and pasting. So very, very simple fake. But it allowed me to generate a lattice. And the point here is having a lattice of islands would be great to do meta-population experiments on the macroscopic scale. But typically you can't, because you don't have that, which will be the point that going to the micro scale allows you to do experiments. But let me introduce island biogeography the way I see it. There's a lot to say about it, but it was really started by these 2 ecologists, E.O. Wilson and MacArthur, at the time at Harvard, where they wrote this book, The Theory About Island Biography. So at the time, there was no island biography. And so the first thing ever published about it was already The Theory of Island Biogeography. And they really started by asking a very simple question: how many species fit on an island? If I get to an island, is there any way of knowing or expecting how many species I should see? Right, that's an emergent quantity that, and it's not clear that in this complex systems you can say anything about it. But they said, well, it's really, perhaps we can think of it as just a competition, the result of a competition between 2 forces. One is kind of immigration that brings species to the shores of these islands, let's say on driftwood, some kind of rodents or other kinds of small animal or insects. And the other force they envisioned to be important is stochastic extinction. So all these populations on an island would be finite. So if you wait long enough, everyone would go extinct. And so by just watching these 2 forces compete, you should be able to create a theory. And well, indeed, they kind of created toy models where you find equilibria, as is indicated here. So the blue line is kind of the immigration rate. The more species you already have on the island, the less is the migration rate. So it has to go down with how many species are already on the island. The red line is the extinction rate. The more species you have, the higher the rate of extinction. And so where these 2 lines cross, they said would be the equilibrium, right? In itself, it's clearly a toy model, but their point was it allows us to discuss trends. Like if you have 2 otherwise equal islands, they just distinguish by size, then the larger one will have less extinctions, and so more species on average. And that had a huge influence, in fact, this book, because people realized we can apply this not only to islands, but any kind of ecosystem that is surrounded by something else. That could be mountaintops, for example, or indeed where it has had the greatest influence was in conservation biology, where people thought about how should we design national parks or natural preserves, because those are also like islands in a way, surrounded by a fence, and so there's nothing coming in or out. And if you want to preserve diversity in there, how should we design it? Well, Jared Diamond, which you might know from the book Guns, Germs, and Steel and other books, well, he had this battle cry, a single large reserve is better than several small reserves, based on this theory. And so really it became influential in even, you know, influencing policies, conservation policies. However, then also criticism was raised, in particular because kind of the tests that could be done were really not very quantitative. I would say qualitative at best, and also they were contradicting one another. And then people also looked, of course, at the theory and said, well, you make a lot of assumptions here, like why should everything be in equilibrium? Why should an island be really homogeneous? And so based on all this criticism, kind of the research dwindled substantially. But I think what remains is certainly conceptual importance here, namely that these emergent phenomena like diversity and evolution and ecology are in principle scale dependent. That was, and so if you change the scale, these phenomena might change dramatically. And that's also, here's a very nice essay, which in the ecology literature makes that point. It's in a way the ecology version of what we have in physics, written by Phil Anderson about emergent phenomena and how kind of basic physics can be found at every new order of magnitude of length scale. As you go higher up in scale, you discover these emergent phenomena which you wouldn't have thought of when you look at the lower scales. And so that is also a famous essay called More is Different. So what I want to do today is essentially I want to develop this island biogeography for microbes on a microscopic scale, and make the point that there actually we can test it. And I'll ask, how does ecology and evolution vary at the microscale? And I'll come back certainly to this one statement that a single large reserve is better than several small. And plus, actually, the benefit here is that for microbes, this is actually potentially also very important, like in terms of health, because like if you ask what's an island for us or another big animal like an elephant, then you need a really big island, right? So for us, a small a patch on smaller than us, 1 square meter patch is not an island. But for bacterium, a little dust particle can already act as an island. And in fact, bacteria see a lot of islands on us and in us. You know probably that we are filled up with many bacteria, as many, roughly speaking, as we have human cells. We have bacterial cells on us and in us, and they often colonize like secluded habitats, like here on the skin. We have sweat glands, sebaceous glands filled with bacteria, and in the gut we have so-called crypts, also filled with bacteria, and they are and these act like islands in a way. We can imagine that at least, and on these islands they live and they kind of propagate and go through generations quite stably. So you can measure this indeed in mice by trying to push out a resident strain by bringing in an invader strain. It turns out to be very hard. That's called colonization resistance. And So that's an important phenomenon that we need to understand if we want to understand diversity, because if there's already one species on an island and it's kind of fending off an invader, that means it's very hard to increase the diversity. So we set out to try to understand that with microfluidics in kind of our hypothesis. If you, it was, perhaps a little bit violent. We call it the Spartan hypothesis, because they, going back like to the Battle of the Thermopylae, were using the topography of their environment to fend off a supreme invader, right? So they famously found kind of a narrow mountain path that allowed them to hunker down and defend these invaders. And they still died in the end, all of them, but they still, managed to do this for a week to fend off these invaders. And the question is, do the bacteria do the same? The experiments I show you are much less violent, but we are about to do also experiments that involve microbial warfare, which in fact exists, which is kind of funny. They have some microbes have chemical daggers to kill other species. We are just going to look at competition, but we also want to see the bacteria and kind of the surrounding topography of the microenvironment to see how colonization is affected. And so this is then the place where Yuya Carita comes in. That's a former fantastic PhD student that I had. He's now a postdoc in Plön at the Max Planck Institute there. And so he created this microfluidic device, which you can see here, which is really just a channel system, has a width on the order of 50 microns. And the basic idea here is we bring in nutrients and bacteria, and then as these bacteria are floating along, they're exposed to these invaginations, and then we can watch them colonize them and potentially evolve. That's kind of the future here. It looks like a pan flute, so we call it the microfluidic pan flute. And we study different kinds of bacterial species in it. Here you see mostly experiments involving this species, Acidobacter species, which comes from the fly guts, which is studied by my colleague Will Luddington. And so in this device, what we see is the following. So as we come from the small cavities and go towards the larger ones, then we see that, well, the small ones are most of the time really empty. There's nothing really growing there. And there's relatively suddenly a start of colonization where these cavities become filled at some characteristic scale, LC, and then increase in density. And so there's a first transition happening here. Of course, the question is, how can we understand that? And this indeed is a competition of two forces. One is there's growth inside these cavities, which increases the density, but there's also stochastic outflow, because these cells, they diffuse randomly, and if they diffuse high enough in these cavities, they get caught by the flow lines of the supply of the flow here in the supply channel, and carries them away. That's like an effective death. And both of these forces scale differently with length, which results in this characteristic scale popping up. You can see this in a very simple reaction diffusion theory shown here, where we describe the density of the bacteria as a function of here vertical position along the cavity and time t, like this. So the rate of change of this density is controlled by two terms. One is diffusion. Sorry, I have something that I have to get out of the way. This control panel, hide video panel. No, this was wrong. Hide this one. Okay. Yeah, one term is diffusion, and the other one is growth. And we have to supply this with boundary conditions. One says that cells are not supposed to leave out of the floor of this cavity, and the other one says, well, the density at some point, let's call that x equal to 0, goes to 0. And from that, you can do a normal mode analysis, for example, and find that there's a critical length scale coming out proportional to the square root of the diffusivity, of the ratio of the diffusivity and growth rate. And when the cavity is too small, you get invariably then extinction. All these normal modes decay exponentially. And if it's larger than this critical length scale, you get exponential growth. And this characteristic scale is, we can figure it out by measuring growth rate and diffusivity separately. It's it's on the right order of magnitude, but what's more important is, like, at the transition, we can also compare the predicted critical density profile, which is just the lowest mode, cosine here, with the kind of integrated density profile that we measure in time-lapse movies, which are these red curves, and it works quite well. So, yeah.
It's asking a question in the chat. So they just said, what is R?
R is the growth rate of the bacteria. And here, right, here it is just a constant. And we measure it to be 0.33 per hour. So it takes about 3 hours to divide.
Mm-hmm. Great.
Any other question? Not yet.
Wait, just the x equals 0, it's like in the figure here, it's that point of that, like that scale that you have here.
Yes, it's where the density goes to 0. Well, we know somewhere it has to go to 0, and that's where we put x equal to 0.
Okay. Yeah.
Sorry, can I just, I guess what I don't understand is, if R goes really high, so like you have really fast growing cells,
Mm-hmm.
I would have expected the channel to fill up, and this suggests that it would be empty. Am I getting that right?
No, if R is very, very large, then indeed this critical length scale goes to 0. For any cavity size, you would then be larger than this critical length scale, and so you would get a filling up.
I see. Cool.
And you raised, well, an important point, that this theory is kind of limited in the sense that as soon as you get a filling up, this theory just predicts exponential growth, right? So it doesn't predict how it saturates, because exponential growth has to stop at some point. That's pretty clear. But this theory is linear, doesn't have a nonlinearity, and so something is missing, and that will be the next part.
Cool.
It's just described. It's kind of a linear stability analysis of the empty cavity, technically speaking. Okay, so let's go and look at the cavities that are larger. Then you see the behavior in this time-lapse movie going about 30 hours. And so you see, as you go to these cavities, as the cavities become larger, the density increases. And that's, you might have expected, you go away from this colonization transition, so it should fill up more. But again, there's something like a transition, because suddenly this dark phase appears. I hope you can see my arrow, where something is denser. In fact, if you do confocal microscopy, you see that the bacteria are actually touching each other. So there's we call it the jamming transition, and this is a rather discontinuous one. And what's going on here is then our next question. And of course, we have to describe these steady state populations now by introducing a nonlinearity. Somehow the question is, where is it? Is it in the growth term, perhaps? That's the natural choice, perhaps, because these densities become large, and you might think nutrients run out, so the growth rate should become smaller. But we can measure the growth rates here in all these phases. It's pretty much the same. So what actually does change is the diffusivity of these bacteria. So they just passively diffuse here. And like colloids, as they become denser, their diffusivity changes. And what effectively then happens is kind of a collective traffic jam in these cavities that flip into this dark phase or coexistence of dark and or jammed and gas-like phase. And there you have these collective traffic jams, which induce this phenomenon, as it turns out. And it's also interesting to actually look at the dynamics, how these phases form. I'm showing this here, where you see that in the large cavities, these phases appear quite rapidly and relax rapidly, but it takes many hours relatively close to the transition to develop this phase and reach equilibrium. It's also kind of a hallmark of kind of critical slowing down near this transition. Now, we can ask on theoretical level how we can describe this. And the first thing is to go back to this reaction diffusion system that I had introduced. As I said, the growth actually hardly changes within error bars. We can't measure a big difference, and the difference is definitely too small to explain what we see. But if you introduce this density-dependent diffusivity, then we can actually show for such a theory, we can solve for the steady state semi-analytically up to an integral. We can determine what the density profile is in the steady state. And the bottom line is that if the diffusivity as a function of density has a negative slope, strong enough, it has to be strong enough, then it can drive a positive feedback, in the sense that if the density fluctuates up, then there's less diffusion, there's less outflow of the cavity, but growth continues, right? And so the density goes up even further, and you get into this vicious cycle, which can only stop when things jam.
Oh, sorry.
Yeah.
There's just a question in the chat. So it says, have you considered the cells that might escape a cavity but get caught in the preceding cavity? So in this condition, the growth rate would be kind of mutual between different cavities.
Sorry, have I considered whether cells that leave one cavity enter another cavity? Is that the idea?
Yeah.
Right. So that I'm sure is happening, but it's a very small, modest effect, because simply the density in the supply channel in terms of bacteria is very small. And in fact, you can model this here by saying the boundary condition for our bacterial density is not 0 at this absorbing boundary, but we say it drops to whatever it is in the supply channel, and the phase diagrams look essentially the same. So that doesn't have a big impact.
Thank you.
It's important for something else that I'll get to later. Okay, so what could generate this negative slope in the diffusivity? Well, if cells are sticky, and they often are, many cells are kind of sticky, or they express an extracellular matrix that is sticky, that of course can plausibly generate clustering and effectively like a negative slope in this diffusivity. So we asked, can we get this even if we have just hard spheres that just repel one another? It turns out, if you figure out what this effective diffusivity is, and we did this in collaboration with my physical chemistry colleague David Limmer, that indeed this diffusivity has a hump-like shape. For small density, it has to increase. In fact, one can show here a lot of things analytically, but for large densities, as you go to random closed packing, it comes down. And this coming down is in fact strong enough to drive a phase transition here, which is illustrated here by plotting the packing fraction at the floor of the cavity as a function of the control parameter, which is kind kind of this length of the population divided by the establishment length, so this critical length upon which you get colonization. So this parameter has to be larger than 1. And what you'll find is kind of a fold bifurcation, that as you increase this control parameter, then at some point the density jumps to the closed packed one where you have jamming. And the density profile also develops kind of a sharp kink here as a function of position. This also predicts hysteresis behavior. So this, which we tested experimentally. I don't know whether the next slide will show this. I can, if there are questions, I can show this experiment, but we can tune this control parameter in different ways. For example, by changing the temperature, so we can go following the red curve and then also go back along the purple curve and demonstrate that the jump happens at different points.
Can I ask you a quick question?
Sure.
Yeah. Hi. So because you have no saturation on the growth rate, so if the system gets jammed, you keep on producing more particles, right? So it's,
yes.
At first sight, it seems that there is nothing preventing exponential growth in this equation. Am I correct, or?
Well, you have a nonlinearity that in principle can and will prevent exponential growth, right? So this here in the diffusivity, you have a nonlinearity that ultimately limits then growth, right? This diffusivity acts then as the way it behaves here. So effectively, it has this hump-like shape, and then when things jam, it kind of shoots up effectively, which means that the density cannot get larger than this random closed packing situation, because if you increase it by a tiny bit, then you get a rapid outflow that just balances the growth, right? So what is balancing growth here is the flow out of the cavity.
Oh, I see. Okay. Yeah, thanks.
One thing, I don't have it here, but one thing I wanted to stress is that some people might be reminded of what's called motility-induced phase separation. This one is a different beast. It requires proliferation, and the diffusivity is always positive here, right? So there's no negative diffusivity arising at this transition. So it's a different thing. But
I'll ask a very quick question.
Yeah.
If you reduce the growth rate by flushing an antibiotic through the channel, will you still be able to get jamming?
Well, actually, I can show this on the next page where we actually do this.
Oh, okay, thanks.
And we did it because, well, in the context where we now want to test whether actually when we take these pan flutes and we pre-colonize them with our wild type strain, can we actually bring in an invader that takes up any of these islands to test for this, what's called colonization resistance. And it turns out if you just generate a sister strain that behaves exactly the same way as the resident, it's just labeled green, then you have no chance. You don't really observe. We don't observe anything within 5 days, which is kind of the maximum time we ran these experiments. And so we have to give the invader an advantage, and the way we do it is to bring in an antibiotic called tetracycline, which slows down growth and ultimately prevents growth. And so the first thing you'll see when I start the movie is the impact of that antibiotic on the resident population. And right now you see below these populations being jammed in these 4 cavities. And so when I bring in the tetracycline, you'll see that this actually dissolves and becomes gas-like, as you can see now. Not everywhere, actually. So the cavity on the left stays jammed, just at a kind of different level. The growth rate overall has reduced, and correspondingly kind of you're closer to the transition here, but it doesn't switch to gas-like. Does that answer your question? So you can still get jamming, just it depends on the action of the antibiotic. If the antibiotic fully stops growth, then all of this goes away. All the cells ultimately behave like colloids. They would just diffuse out of these cavities and, well, homogenize across the whole system. So you need growth. It's kind of the engine for this non-equilibrium phenomenon here. And but if you can use to some extent antibiotics to tune the system. Okay, so now let's look at these green invaders. So after 24 hours, there's a first little perhaps invasion right here. Let's look at the next time, the next day. If I run this movie, you'll see some successful invasions have occurred here, for example, in this cavity. And after 5 days, the situation really depends strongly on the cavity you're looking at. Some cavities have very little invasion, some have a lot. And so this distribution indicates there's really now kind of correlation between these cavities. So we believe, even though we haven't shown it yet, that if you get some first invasion, this kind of acts as a source for more invaders locally, and then that leads to kind of an expansion of the invading population. But importantly, we never see any invasion of these jammed populations. So here you see jammed populations, they're uninvaded. Likewise here, in all cases, these jammed ones, they resist any kind of invasion. And you can show this also theoretically, that this is practically impossible and would take forever. The reason being that an invader has to be really very successful. It has to infiltrate this dense population and diffuse up all the way to, in fact, the floor of the cavity, or here the ceiling of the cavity, which is just not possible because the diffusivity there things are jammed. This is a glass-like system where the invader cannot really diffuse. It's just super slow. And we can measure these diffusivities also, compare them here by sparsely labeling cells. We can measure them in the gas-like state, and we also in this jammed state. It's hard to track individual cells, but we can co-culture them with the unlabeled and labeled strain, and then look at the fluctuations of these emerging boundaries, which gives us a clue of what the effective diffusivity is there. It's not zero because of all the growth, right? The growth effectively melts this glass-like system and generates some diffusivity, but it's very, very slow, very low. Okay, so that explains essentially that indeed here these jammed populations cannot really be invaded. And I'm almost here at the end with what I wanted to say. I just wanted to show you another experiment which looks essentially at one part that's relevant for evolutionary dynamics, which we are now looking at, like in these cavities. We want to understand how populations evolve and how it depends on kind of the ecological phase they are in. What we did here is, rather than bringing the invader from the outside, so first we pre-colonized it. In the previous experiments, we pre-colonized the pan flute and then brought in the invader from the outside. Here, we co-cultured them at time 0. We brought them in at the same time, and then see what kind of populations develop. And we see frequently these mixed cultures where you have kind of a sharp boundary between dark and labeled, which one can show that this has to be the case. But then we turn on the antibiotic and should observe how this resistant strain, green, should take over. And it does that, but kind of in an interesting way, which you see in this movie. Let me do it again. I'll show it again. There are kind of two phases that are happening here, which I find interesting. The first one is this one here, where even though the antibiotic just affects the dark strain, you also lose part of the green strain. In terms of physics, we can understand that because the green strain depends to some extent on the dark strain for not leaving the cavity, right? The dark strain helps to jam up this population. So if you remove in a targeted way only dark cells, you also mobilize the green ones and they leave. So there's really no advantage in this phase for the green population. But only once the population turns into effectively a gas, then you get exponential takeover by the green strains, which I find quite interesting because how effective kind of the selection for resistance is depends on the ecological state. And it's more effective if kind of the total population size is smaller. All right, so my conclusions would be that, well, let's go back to this battle cry of island biogeography. A single large reserve is better than several small reserves. Certainly not generally true in our pan-flutes because it depends. What's the ecological state of the population? Because certainly in these jam populations, they're resistant to bringing in any additional strain. So essentially, they are filled up if one strain is already present. So very strong colonization resistance there. But in the gas-like populations, perhaps, right? But generally, there is a trend as you increase the islands, these cavities, they tend to jam up. We think this also has implications for like the actual situation where bacteria colonize little cavities on us and in us, because you can imagine that over evolution, we as a host for these bacteria perhaps have shaped our cavities in order to enrich for the bacteria we are interested in, right? This provides you a mechanism that tells you, you can indeed, by changing the shape and size of your cavities, you can make it more or less habitable for a given bacterium. And in reverse, bacteria might also adapt to the cavities they find. So I find that a very interesting possibility. Of course, we have no proof for that. There is a trade-off between stable colonization and competition, which I showed you here in the end. And generally, near the phase transitions that we observed here, there's an extreme scale sensitivity, which makes it necessary, we think, to study kind of the combination of ecology and evolution on multiple scales and vary the scales and the shape of these cavities. All right, so with that, let me thank Yuya, who did most of this work, but he's standing on the shoulders and here actually the heads of former group members who really helped in many ways shape this project. And here papers if you're interested in this, and I'm looking for postdocs, so please reach out if you're interested. Thanks for listening.